We're going to continue on with the nervous system. So 37.2 is all about the generation of action potentials and how those action potentials get generated by the movement of sodium and potassium ions. So the inside of a cell is negatively charged relative to the outside of the cell. This difference is a source of potential energy, and it's called membrane potential. The resting potential is the membrane potential of a neuron that's not sending signals. Changes in membrane potential act as signals, transmitting and processing information. So basically when the membrane potential changes, this allows neuron cells to generate action potentials, in a sense, fire. They generate electrical impulses down their axons. Potassium and sodium play an essential role in forming the resting potential. In most neurons, the concentration of potassium is highest inside the cell, while the concentration of sodium is highest outside the cell. As you recall, sodium-potassium pumps use the energy of ATP to maintain these potassium and sodium gradients across the cell. So as you recall, 3 sodiums get pumped outside the cell, and 2 potassiums get pumped inside the cell. So your neuron cells, your nerve cells, are pumping 3 positive charges out and 2 positive charges in. Part of the reason why the inside of the cell is more negative than the outside of the cell. So here you see some ion concentrations both inside and outside of mammalian cells. You can see that there's a very high concentration of potassium inside the cell, okay, as a result of the sodium-potassium pump pumping 2 potassiums inside the cell. You can see that the sodium concentration is very high outside of the cell. Sodium-potassium pump pumps 3 sodiums outside of the cell. And then you have other ions which contribute to charge, like chloride and other large anions, which are negatively charged ions. Okay, so basis of membrane potential. So here we have the sodium-potassium pump. So the circles are the sodiums. So you can see that the sodium-potassium pump is pumping 3 sodiums out for every 2 potassiums that come in. Now there are also channels within the membrane, both sodium and potassium channels within the membrane. So you can see that potassium is going to diffuse down its concentration gradient out, and sodium is going to diffuse down its concentration gradient into the cell. The opening of ion channels in the plasma membrane converts the chemical potential energy of the ion gradients to electrical potential energy. Ion channels are selectively permeable, allowing only certain ions to pass through. A resting neuron has many open potassium channels, allowing potassium to flow out. As pictured here, you see many more potassium channels as opposed to sodium channels. So another reason why the inside of the cell is more negative than the outside, because you've got all these potassium ions that are leaving the cell. The resulting buildup of negative charge within the neuron is the major source of membrane potential. The equilibrium potential is the membrane voltage for a particular ion at equilibrium and can be calculated using the Nernst equation. The equilibrium potential for potassium is negative 90 millivolts. The resting potential of an actual neuron is about negative 60 to negative 80 millivolts because of a small amount of sodium ions that diffuse into the cells. Okay, so how are action potentials generated by your neurons? Researchers can record the changes in membrane potential when a neuron responds to a stimulus. Changes in membrane potential occur because neurons contain gated ion channels. Gated ion channels are channels that can be opened and closed in response to a stimulus. So here you can see the first gated ion channel rather, is closed. So these ions are not getting into the cell. A change in membrane potential or voltage across the membrane can cause a gated ion channel to open, allowing, in this case, the influx of ions into the cell. Okay, there can be hyperpolarizations and there can be depolarizations of the membrane. When gated potassium channels open, potassium diffuses out, making the inside of the cell more negative. This is hyperpolarization, which is an increase in magnitude of the membrane potential. Okay, so remember that your resting potential of your membrane is about between -60 to -80 millivolts. So if more potassium continues to leave the cell, then that resting potential becomes even more negative. So hyperpolarizations are when the resting potential gets further away from what we call a threshold. Okay, so this is a graph looking at the resting potential of a neuron. Okay, so here's the resting potential between -60 and -80 millivolts. There's some sort of stimulus that causes an increased permeability to potassium. So potassium will leave the cell, making the cell more negative. So here you can see that it becomes more negative. That's called a hyperpolarization. It brings the membrane further away from what we call threshold. And we'll get to what threshold is in a minute. Opening other types of ion channels triggers depolarization, a reduction in the magnitude of the membrane potential. For example, depolarization occurs if gated sodium channels open and sodium diffuses into the cell. Okay. So if we look at that, so here's my threshold. This is a magic number that you'll know the significance of that in a couple minutes. So in this case, you have a stimulus that increases membrane permeability to sodium. So sodium will go into the cell. Sodium is positively charged, so when it goes into the cell, it makes the membrane potential less negative. Okay, so we are approaching this threshold number. Graded potentials are changes in polarization where the magnitude of the change varies with the strength of the stimulus. Graded potentials decay with distance from the source. If a depolarization shifts the membrane potential sufficiently, it results in a massive change in membrane voltage called an action potential. Action potentials have a constant magnitude and transmit signals over long distances. They arise because some ion channels are voltage gated, opening or closing when the membrane potential passes a certain level. Action potentials occur whenever a depolarization, remember depolarization, membrane becomes less negative. So whenever a depolarization increases the membrane potential to a particular value called the threshold. Action potentials are all or none. Okay, so what does that mean? So here's our graph again. Here's the resting potential between -60 and -80 millivolts. You have some sort of stimulus. That stimulus causes the membrane potential to change. Okay, so we are going closer to this magic number, which is the threshold. Now, once this threshold, this magic number gets reached, then an action potential will be generated. That's what's meant by all or none. Once we reach threshold, your neuron will fire. It's an all or none response. Either it's going to reach threshold and it's going to fire and generate an action potential, or it's not going to reach threshold and it's not going to generate an action potential. Okay, so we're going to talk next about what causes this change in membrane potential. An action potential can be considered as a series of stages. At resting potential, most voltage-gated sodium ion channels are closed. Most of the voltage-gated potassium channels are also closed. Okay, so this is a very handy diagram as to what's happening, and it relates it back to that graph that we just looked at. Okay, so here is the resting state of your neuron, of your end of your membrane. Okay, inside of the cell is more negative than outside. You have voltage-gated ion channels that are closed. Then you have a stimulus which will depolarize the membrane. Some gated sodium channels open first and sodium flows into the cell. During the rising phase, the threshold is crossed and the membrane potential increases. This is also called depolarization. During the falling phase, also called repolarization, voltage-gated sodium channels become inactivated. Voltage-gated potassium channels open, and potassium flows out of the cell. Okay, so here we have depolarization. Your sodium-gated ion channels start to open and sodium goes into the cell. This causes the inside of the cell to become less negative. This is sort of like a positive feedback thing where once sodium starts to go into the cell and that threshold gets reached, more sodium channels open up and more sodium goes into the cell. Okay, so that was the first part of the graph where the line goes up and it reaches the very top. Okay, so that's the rising phase of the action potential. And then once the action potential gets generated, your sodium channels will close and your potassium channels will open. So the purpose of opening your potassium channels is so that potassium can leave the cell. Now when potassium leaves the cell, you have negative charges leaving the cell. Positive charges leaving the cell. That allows the cell to go back to, and the membrane to go back to its resting potential, where the inside of the cell is more negative than the outside. Okay, so it maintains or reestablishes rather that resting potential. During the undershoot, membrane permeability to potassium is at first higher than at rest, and then voltage-gated potassium channels close and resting potential is restored. Okay, so here's your undershoot where your potassium channels stay open. They have increased permeability, but eventually they will close and you will level back off at the resting potential. So looking at this graph, let's just go over again what's happening at each step. So step 1 is our resting potential. Resting potential between negative 60 and negative 80 millivolts. Some stimulus causes voltage-gated sodium channels to open. Sodium starts to move into the cell. We reach threshold. When we reach threshold, more sodium-gated ion channels open and sodium rushes into the cell. This causes the generation of an action potential, causes your neuron to fire. Once the action potential has been generated, your sodium-gated ion channels will close, your potassium channels will open, and potassium will leave the cell, causing the membrane potential to go back to its resting potential. This right here is called the undershoot, and this is just when the cell is more permeable to potassium than it is when it's at rest. Okay, but eventually it levels back out at the resting potential. Phase 3 is called the rising phase or the depolarization phase, and phase 4 is called the falling phase or repolarization. During the refractory period after an action potential, a second action potential cannot be initiated. The refractory period is a result of a temporary inactivation of the sodium channels. For most neurons, the interval between the start of an action potential and the end of the refractory period is only 1 to 2 milliseconds. At the site where the action potential is initiated, which is the axon hillock, an electrical current depolarizes the neighboring region of the axon membrane. Action potentials travel only toward the synaptic terminals because of this. Inactivated sodium channels behind the zone of depolarization prevent the action potential from traveling backwards. Okay, so here's what that looks like. Okay, so you have your sodium channels which are open, which are causing the action potential. So the action potential gets generated here at the axon hillock. So it fires, and then as it fires, just behind that, the sodium channels close and the potassium ones are open. So that's why the action potential gets generated in one direction from the axon hillock down to the synaptic terminals. Now there's some adaptations in axon structure that increase the speed of the electrical impulse. So the speed of an action potential increases with the axon's diameter. In vertebrates, axons are insulated by a myelin sheath, which enables fast conduction of action potentials. Myelin sheaths are produced by glia. Remember, those are the supporting cells of the nervous system. Specifically oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. I know probably all of you have heard of multiple sclerosis. Multiple sclerosis is an autoimmune disease where the cells of your immune system actually attack the myelin sheath of your axons, of your neurons. So it sort of, destroys those cells. So it, decreases the efficiency of your axons to be able to fire. So that's a big problem. Very serious disease. Okay, so here, this again is your axon. These are your Schwann cells, and the spaces in between your Schwann cells are called the nodes of Ranvier. And basically what is happening here, the myelin sheath, the reason why the electrical impulse fires so quickly is because it actually jumps from one node of Ranvier to the next, to the next, to the next. This is something called saltatory conduction. Action potentials are formed only at nodes of Ranvier, gaps in the myelin sheath, where voltage-gated sodium channels are found. Action potentials in myelinated axons jump between the nodes of Ranvier in a process called saltatory conduction. A selective advantage of myelination is space efficiency. Okay, so here's a picture of that. Here's your axon hillock, where the action potential gets generated, and then it jumps from node to node. Okay, and that allows the electrical impulse to fire very quickly. Okay. We're going to stop there.